Hello student, welcome to another episode of Knowledge Quest. The subject is biology and I'm your teacher Lydia Asiago. Today I want us to walk our mind into a market. Okay? Are we ready? We go to a market? Yes, I'm ready to take you to a market. Now, when you walk into the market and probably you want to get some of the vegetables for your meal, what do? For example, today I feel like taking some sukuma wiki, some kales. So when I go into the market, I make a walk. As I go around, I try to check which one is the best. Then I meet one of the sellers with a lot of vegetables that if I try to look at these vegetables, they are not appealing. They are not appealing. So I look aside and continue having a walk. I walk to another seller. I find another seller having a bunch of kales that are placed in a bucket, that bucket has some water. So I try to look at these kales and I see the kales, they look fresh, they look nice. Then I try to ask myself, the first seller had some kales, and those kales, they looked that if you look at them, they were not appealing. Then I asked myself, what do you think is wrong with those kales? Why do you think the kales that this farmer had on the table, probably where they are selling, they are different from the ones that someone else had some kales, but these kales were placed in a bucket of water, and then they looked very fresh and they were very appealing. As I was doing another walk, still on the same, I found another farmer, probably a seller, and this seller, she was a lady. I found her taking the kales, and these kales, she took a thread, she tied around the kales. Okay, I'm trying to demonstrate what this seller was doing. She was a lady. Then she took the kales, she had a bucket of water, and she placed the kales in water. Then I could see her removing it from the water and placing it aside, probably waiting for the what? For the buyer. All this reminded me of osmosis. So today, we want to learn about osmosis. What is the role of osmosis in plants? And we are going to pick one of the subheading of osmosis and what does osmosis do in plants. So, in our lesson today, we are going to learn biology, and our topic for today is role of osmosis in plants. What is the role of osmosis in plants? And particularly, what is the role of osmosis in relation to support in plants? Of course, I understand all these sellers, they had an idea of what they're doing. Probably at one time they were in class. And I want to believe the seller who had the kales in a bucket of water and the seller who took the kales and placed in a bucket of water, I want to believe they were applying their concept, the concept of osmosis that they learned in their while they were in school. So today I want us to explore more and at least try to understand. We want to understand why that you have some kales, you place them in water, others, they are not in water, and you find that most of them, especially if you go to the market, most of those sellers who are selling vegetables, they can't miss a bucket of water. Most of the farmers or most of the sellers, those who are in the market and they have their vegetables that they want to sell, mostly you find that they have a bucket of water besides them. So today I want us to understand more, why do you think they must have a bucket of water besides them? Now, when we talk about osmosis, I want us to understand the definition of osmosis. I want us to understand the definition of osmosis. Osmosis is one of the physiological processes that takes place in living organisms. So, if I was to define osmosis, I'll say osmosis is the process, okay? Osmosis is the process by which water molecules, we say water molecules, they move from a highly, we say water molecules, they move from a region where there is high water molecules. So maybe we can use osmosis, we can define it in 3 ways, but this time I want to use this one. Osmosis is the process by which water molecules, they move from hypotonic solution to hypertonic solution. Okay, through a semi-permeable membrane. So we say through a semi-permeable membrane. Okay, so we want to understand the concept of osmosis in regard to what our sellers do in the market. So we say osmosis is a process by which water molecules, they move from hypotonic solution to a hypertonic solution through a semi-permeable membrane. Now, when we talk about hypertonic solution and hypotonic solution, an example, so I can give you an example of hypotonic solution. Hypotonic, we have distilled water, so we can have distilled water. Okay, so distilled water is an example of hypotonic solution. Okay, remember distilled water has more water molecules. We can use another example of hypertonic solution. Hypertonic solution as salt solution. Okay, so hypertonic. So those are just examples of solutions that give us different regions. So we talk about osmosis is the process whereby water molecules, so in this case, it is the water molecules that move from a region where you have a higher water concentration to a region where you have a low water concentration. And basically, osmosis is the reason or the role behind the support that we have in plants. So I want to demonstrate that. So we are going to demonstrate that so that we understand why these leaves that are placed in the open air. So for those sellers who have their kales or their leaves or their vegetables placed in the open place, you find that their vegetables, they look like they are flaccid, they look weak, and if you check on the cells. If you check on the strength, you find that the plants look like they are very weak. They cannot even be upright. But if you talk about the leaves or the scales that are placed in a solution or in distilled water, you find that the leaves are very upright. The leaves are upright, okay? And then you find that the leaves, even the stalk, this part is very firm, unlike this one, that is very what? Very weak. So this one looks very weak, but this one looks very, very firm. And of course, the leaves, this one, the leaves, they are very upright, but this one, you see, the leaves are very flaccid. Usually we say they droop. Now, I want us to see practically what happens that if you place, leaves or these vegetables in water, there is a difference than those that are exposed to the air. Now, with me, I'm going to demonstrate so that we have the two solutions, that is the two conditions whereby we have hypotonic and hypertonic solution. So with me, I have salt. So we say this is common salt, okay? This is common salt. So I want to make a solution of salt solution. Common salt, we also call it sodium chloride, okay? This is sodium chloride, which is the common table salt. Now, I want to make a solution of sodium chloride. So I'm going to take a beaker, and in this beaker, I'm going to label. Remember, we always say that when you carry out experiment, you always have to label the reagents or anything that you carry out, you need to label to avoid confusion. So here I'm going to have salt solution. So I'm going to have salt, okay? So this will be salt solution. Is that okay? Yes, so I'll get salt solution here. So that in this salt solution, for me to prepare the salt solution, I'm going to have a pinch of the salt, and I place it in a beaker, okay? So I'm placing some salt. Now this salt, I'm going to add water so that I prepare salt solution. I will add some water. Then I'll try to stir. Remember, I'm preparing salt solution to represent a region of a higher concentration, a hypertonic solution. And we said hypertonic solution, of course, it is a solution that has more solute molecules and less solvent molecules. In any solution, usually we have a solute and a solvent. So a solute, in my case, I've used salt, and the solvent, in my case, I've used water. So water is the solvent in my case. So, and in that case, now I have prepared salt solution. So I'll put it aside, and then I have another setup whereby I'm going to have distilled water to represent hypotonic solution. So this one I will use distilled water, okay? So this I will use distilled water, and then I place it in the beaker, okay? Remember, I'm saying I want us to understand why do you find that most sellers in the market, they sell their vegetables, but they can't miss a bucket of water besides them. Now what happens in osmosis, so I'm going to take the two setup. This farmer, probably this seller, has the leaves that look very weak. The they look very weak. So I will use both cases. I will get a sample from the scales that are left in the open air. Then I place them in the two solutions. I'll also get another specimen from the scales that are placed in distilled water, that is in water. Then we are going to get the difference. What will be the difference? So because I want to have equal amounts so that we are able to make the observations. Remember, observations, we can make observations either through sight, what we see, through touch, what we feel, because we can use our senses to make the observation. So I'm going to cut pieces of the petiole. So this one, we call it the leaf petiole. Leaf petiole. So I've used the petiole of a sukuma wiki, and I want to cut equal pieces. I only need 2. Okay, I only need 2, so I'll take 2 pieces and I cut. Okay. So because I want equal pieces so that I'm able to take the measurements at the end, then I will do, that is 2.5 centimeters cubic. So I have 2. Remember, these are petioles from the sukuma wiki that were exposed in the open. The sukuma wiki that looked very weak, the leaves are very weak. We say they are flaccid. So I place in both the solutions. Then I take another one. I also take another scale and I cut the petiole. I'm going to cut the same equal amount of the petiole. So for me to ensure that I'm accurate, that's why I'm using a ruler. So I'm using a ruler. I want to measure. I also have equal amount. So I will cut it at 2.5 centimeters. Okay, that is at that point. So at that point, I still have 2 pieces. So I'm going to place this specimen still each into each solution, so that now we leave the setup. We make observations after some time. But something I want you to remember, these are scales, both are scales. The only difference is this one is exposed to the air. This one, yes, it is exposed to the air, but the petiole is inserted in water. So it means if you were to feel, if you were to make observation by feeling, you could get there is a difference. The leaves that are exposed to the air, if you look at the stem, the part of the stalk, we say that it is very soft, it is flaccid, okay? But the one that is placed in water, if you press on it, it is very hard, very hard. And of course, at that point, we say the cells probably, they are very turgid. So I want us, as we wait for the setups, we waited for a few minutes so that we confirm if we place the specimen from this, the one that was exposed in the air and the one that was exposed, that was placed in water, if we put them in the same solution, will they behave the same or will they behave different? Having in mind that we are discussing the role of osmosis in plants. So as we wait for the setups, I want you to remember of this. In plants, they are made up of, if you talk about plant, a plant is a living organism, and usually we say plants, we talk about plants, so this one we are discussing about plants. So when we talk about plants, we want to look at the cells. When we talk about a plant cell, what is so unique with a plant cell? We realize that a plant cell has a cell membrane and it has a cell wall, unlike an animal cell. An animal cell has only the cell membrane. But you realize that for a plant cell, it has the cell membrane. At the same time, it has the cell wall. So if I was to do a sketch, so I want to assume I'm doing a sketch of a plant cell. Remember, we say a plant cell has a definite shape. A plant cell has a definite shape. So if I was to draw a plant cell, I can do a sketch. Then I can say this is the cell. This is the cell wall. Then this one is the cell membrane. Okay? That is the cell wall. And then we have the cell membrane. Remember, a plant cell has another membrane that we call the tonoplast. So inside here, we have another membrane. This membrane, we call it a tonoplast. So I want to do, that is just a sketch. Remember, it is a sketch. And probably this is where we have the nucleus. Okay, let me say this is the nucleus. Then have another membrane we call the tonoplast. Remember, I'm trying to draw the structure of a plant cell so that we are able to understand what is happening with these plants. So inside here, this is where we have the cytoplasm. And remember, in the cytoplasm, this is where we have cell organelles. So this is the cytoplasm. And in the cytoplasm, we have cell organelles like the chloroplast. Let me just do a simple structure of a chloroplast. So a chloroplast, this is where we have so if I was to label that, I'll say this is the chloroplast. And remember, plants, they have numerous chloroplasts. Then I'm now interested on this part. I think you need to see that. I'm now interested on this part. Inside here, this is where we have the sap vacuole. Okay? This is what we call the sap vacuole. Okay? So probably I will write it the sap vacuole. So that is where we have the sap vacuole. Now, in the sap vacuole, this is where a plant stores salts and sugars. That is where a plant stores salts and sugars. And mostly, the concentration inside the sap vacuole is the one that determines if a plant cell will lose water to the surrounding or if a plant cell will gain water from the surrounding. And remember, when a plant cell loses water to the surrounding, usually the cells become flaccid. But when a plant cell gains water from the surrounding, the cells become turgid. Now, as we explore that and explain it into details, we are taking a short break. We will be back shortly. Dear student, welcome back. Before we went for a break, we were discussing on osmosis, and we said that in plants, osmosis plays a role in support. And basically that is what you are doing, and that's what you've demonstrated. Now we realize that in a plant cell, just as I've drawn it here, A plant cell has the cell wall, has the cell membrane, has the tonoplast. Then, of course, we have the nucleus, we have the cytoplasm, the sap vacuole. And our interest for today's lesson, we are going to concentrate on the sap vacuole. Now, when you talk about the sap vacuole, you realize that inside the sap vacuole, this is where a plant stores some salts and sugars. A plant stores some salts and sugars. So we want to explain exactly what happens that you find some plants, the cells, they are turgid, the leaves are upright, but in some plants you find that the leaves are very flaccid and the cell becomes flaccid. Basically, you want to discuss why some plants they look flaccid, others they look turgid, and you find out that some leaves they droop in some plants. So you want to explain that using a plant cell. Now I want to draw a simple structure of a plant cell. I want to assume this is a plant cell. This is just a sketch. Remember, it is a sketch of a plant cell. And then we said inside here we have what we call the sap vacuole. So I'm interested with the sap vacuole. Okay, we are interested with the sap vacuole. Now what happens inside the sap vacuole? We have salts and we have sugars. Now these salts and sugars, when they dissolve in water, they make salt solution or sugar solution. So basically they make either a hypotonic solution or a hypertonic solution. So inside the sap vacuole, because we said this is the tonoplast, what happens if you have more salts? If you have more salts, so you have more salts in the sap vacuole, what happens inside the sap vacuole? It becomes hypertonic. Inside the sap vacuole becomes hypertonic. Now, when the sap vacuole is hypertonic, when the concentration inside the sap vacuole is hypertonic, what happens? What happens is this plant, now the plant cell, remember we are talking about a plant cell, the plant cell now draws in water. Remember, this plant cell has the cell wall. Okay, this plant cell has the cell membrane. And what we know about the cell wall is that the cell wall is permeable. Permeable means it allows all substances to move in and out of the cell. And then the cell membrane, usually we say it is semi-permeable. So it allows some substances to move in and out of the cell. So when this sap vacuole is hypertonic, you expect the plant to draw in water by osmosis. So we expect water to get in by osmosis. Now the water gets in all the way to the sap vacuole. And as water gets in, so this is water, is drawn in by osmosis. Okay? Water is drawn in by osmosis. So as water gets into this sap vacuole, what happens is that there is the tonoplast. We expect the contents to increase. As the contents increase, inside here it develops what we call a pressure that is known as Tagger pressure. Now this Tagger pressure causes this cell to stretch such that if I was to draw this, so let me assume I'm drawing this. So we expect the contents in the sap vacuole to increase. So we have the concentration here, and we have the Tagger pressure. This is what we call the tonoplast. So we have the tonoplast. Okay, So here where we have the tonoplast, here we have the what? The cytoplasm. Now what happens as the content inside the sub-vacuole increases, it creates a pressure that we call Tagger pressure. Now what happens is that as this tonoplast, as the contents are increasing, they are creating they press on the cytoplasm, which further presses the cell membrane to the cell wall. Now remember, something that is unique is that the cell wall usually is firm. This cell wall is, so this is the cell wall. So we say we have a cell wall, okay? This is a cell wall. Now the cell wall, usually we say it is firm, okay? This one we say it is firm. It is always rigid. Rigid means it doesn't move. Why? Because the cell wall is made up of the cellulose. So we expect from the two setups, we expect if inside the sub-vacuole it is hypertonic, then we expect that cell to draw in water, and the cell, we expect it to distend. As it does that, you realize that the cell wall also creates a force. So the cell wall also creates a force that is equal and opposite to Tagger pressure, and that force is what we call wall. So we have wall pressure, okay? But at the same time, we have Tagger pressure that is forcing, is extending, causing the contents inside the sub-vacuole to increase. So basically, what makes the cells firm and rigid, firm and turgid, is because those cells are filled with water. Now, maybe from the setup that we had, remember, we had two setups, whereby we have the leaves that had wilt. Let me say, of course, wilting is a process whereby a plant cell, the rate at which this plant cell loses water is higher than the rate at which it absorbs water. So you find that the cells, they end up becoming flaccid. So we took a petiole. We took a petiole from this specimen. We also took another petiole from the other specimen that were placed in water. This one was placed in the open place. This was placed in the water. As much as we said this one, the petiole, the cells looked very tough, okay, hard. Even if you press, they are very hard. But this one, it was very soft. This one was very soft. But we took the two specimen and we placed in two different solutions. So that's what I want us to find out. Did they take the same shape or did they change? So I will start from the specimens that I placed in salt solution. So I'll take all of them, okay? So I have this one. So we want to see if there is a difference. Both are in salt solution. And we want to make observation. Our observation, we are going to base it more on the sense of touch. Okay? Now, if I can touch and feel, I think this is something that you can do even at home. If you touch and try to press, if you press on the two specimens, you realize that both of them, they are soft. Both of them are soft. Why do you think both of them are soft, yet it is the one that was in the open that was soft, yet the one that was in the what? In water, it was very firm. You realize that these two were placed in hypotonic solution. Remember, salt solution, we said salt solution represents a hypertonic solution, okay? So, salt solution. So the specimen the specimen in the salt solution, if I was to record the observation, I can say they are very flaccid. They are very soft. Okay? The specimens are flaccid. They are very soft. Okay? Then I want to see the specimen that was placed in distilled water. Then from there, we are going to explain why do you think these ones are soft? Why do you think they are what? Yet, remember there is one that was what? Hard, but now it is soft. And bearing in mind that these petioles, they are covered by the epidermis, they are covered by the cuticle. But remember, we have the part that the cells are open, the inner cells are exposed. So those are the two specimens that were in the salt solution. I want to pick the others that were in the what? In the, okay, so we'll get the ones that are in distilled water. Okay. If I try to feel them, both of them are hard. The specimens are hard, very hard, tough, okay? When you press on them, they feel very hard. Remember, we had one specimen from the leaves that were exposed in the open, which was soft and flaccid, but now we have placed it in distilled water. It is now hard. That one tells you something. The specimen that we obtained from the one that was dipped in the water, it is still hard. So basically, both of them are what? They are hard. Now, how can we explain that? So the specimen in distilled water, I've said it looks very hard. Okay. Probably we didn't measure the length. We also need to confirm, did the length of these specimens increase? Yes or no? Did they increase? What do you think? Should they increase or decrease? Which one should increase and which one should decrease? Now, maybe first we explain why this specimen that was placed in distilled water, why does it become firm and turgid, and why the one that was placed in salt solution becomes flaccid? So in this case, I want you to have this in mind, that you have a salt solution in one end, and in the other end, you have distilled water. And we said the salt solution, in this case, is representing what? Is representing hypertonic solution. Okay? And distilled water, in this case, is representing hypotonic solution. Okay? And if now you have the two cells, you realize that a cell that is in hypertonic solution, number one, the cell loses water by osmosis. Okay? The cell loses water by osmosis. Why is the cell losing water? Remember, water molecules, they move from a region of a higher water concentration to a region of low water concentration. Now, will this, from hypertonic solution, we expect the cell to lose water, or do we expect the cell to gain water? Now, in this end, because water moves from where it is more to where it is less, then we expect in that case the cells. So this is the cell. And then we have the sap vacuole. So in this case, we expect this cell to do what? To lose water. Why is it losing water? Because the surrounding is what? The surrounding is hypertonic. So the cell will lose water by osmosis. So it will lose, so this will lose water. Okay? The cell will lose water. And when this cell loses water, what do we expect? When this cell loses water, what do we expect? We expect the cell sap now to do what? To decrease. The content in the cell sap will decrease. At the same time, we expect this cell membrane, okay? We expect the cell membrane to detach from the cell wall. So if I was to draw that, I will draw it like this. Okay. So we expect the cell wall to remain intact, but the cell membrane, so that will be now the cell membrane, okay? The cell membrane detaches from the cell wall. Then, of course, we have the tonoplast. So here we have the tonoplast. Here we have the cell wall. Here we have the cell membrane. Okay? Here we have the cell membrane. Then what is happening is that the cell membrane detaches itself from the cell wall. Remember, when it is stretched to the cell wall, that is when the cell is filled with water. But this time, the cell loses water by osmosis. And we say the cell become flaccid. Okay? The cell become flaccid. Why? It has lost. So the cell membrane tries to detach itself from the cell wall. And of course, the osmotic pressure in that case is very low. But what happens when this cell was placed in distilled water? Distilled water, we say the solution now becomes hypotonic. There is more water. So what happens? This cell will draw in water by osmosis. Remember, I'm trying to draw the cell of a plant which has the cell wall. Okay? Then here we have the cell membrane. Okay? Now in this case, now we expect the cell to do what? To draw in water, and the tonoplast now will swell because there is a lot of water in the what? In the cell. So at this point, if you are very keen, you will see that here, this is where we have the sap vacuole, and then this is the cell membrane, this is the tonoplast. And then inside here we have the sap vacuole. So this is where we have the sap vacuole. Then also inside here we have the sap vacuole. Okay? Now in this case, what is happening? Number one, you realize that if you place a plant cell in distilled water, the plant cell does what? Gains water by osmosis. Okay? The cells gain water by osmosis. Why? It is because inside, when outside the environment of the cell outside is hypotonic, then it means it is hypotonic to the sap vacuole. So that means the sap vacuole is hypertonic. Being hypertonic, it will draw in water. So this one draws in water. So we say draws in water. But this one, the cell that was placed in hypertonic solution, what happened? It lost water. So this one, it lost water. And that is why we are saying this cell is what? Flaccid. It has lost water, so the contents inside the cell, that pressure that usually builds within the cell is very low. So here, the target pressure is very low, but here we say the target pressure is very high, and that's why here now we'll say the cell become turgid. The cell become turgid. So in those two cases, we are saying that plants need what? Plants need water. The cells of the plants, they need to be turgid. That's why we are saying the role of osmosis in plants, especially in the part of support. What happens? If you talk about the herbaceous plants, plants that do not have a woody stem, what happens? How do they attain their support? Plants that do not have a woody stem, their stem is not hard, their stem is soft, their stem is succulent. It is because they have parenchyma cells. Those parenchyma cells, they get filled with water. As they get filled with water, the cells become turgid. So indirectly, that turgidity makes the cell firm, provides the cell with support. It's a mechanical support. So in other words, we are saying, for those sellers, as we walked with you into the market, remember we started by walking our minds into the market, and we saw two sellers. One was selling vegetables that were just in the open. One was selling vegetables that were placed in water. And the other one was having vegetables that were placed in water, but now they were placed somewhere. Now, I also need you to understand, as we learn this concept, remember, the leaves are covered by the cuticle, okay? The leaves are covered by the cuticle. The cuticle is waterproof. The cuticle is waterproof. Does not allow water loss or water gain from the surrounding. Plants can only get water from there through the what? Through the xylem. It is the xylem that is in the plant that transports water. So what happens? This plant is not drawing in water from the leaf. It is drawing from the petiole here. If you were to cut the petiole and you expose this part, what happens is that this part of the petiole, it has what we call the xylem. So it is the xylem that is in this petiole that is transporting water from any source. Probably if you are placing your specimens in water, water moves from the what? The xylem. So it is the xylem that is getting water from it is the xylem that is getting water from the source now as it transports it into the leaf. Remember, the farmer who places, or the seller who places the vegetables in water, it does not mean that the water gets direct into the leaf. No. This seller just wants to get the surrounding of the cells, the surrounding of the leaves, to be moist. In a way, he's trying to reduce the diffusion gradient. The rate at transpirational, the diffusion gradient, the transpirational, plants lose water through transpiration. So in a way, it's trying to reduce the saturation deficit so that less water is lost to the surrounding. So water does not get direct into the leaves, but water gets through the exposed part where we have the cells, and water is transported all the way to the leaf. My student, I hope you've tried to work with me and try to understand why most of the vegetables, they remain upright, the leaves are upright, the leaves are turgid when they are placed in water rather than when they are placed in the open place. Thank you for watching our episode. Meet you in the next episode. Keep tuning to our channel, Hope Channel Kenya, and you learn more. I've been your teacher, Lydia Asiago.